Oriana, tell me now it's time. It is time. So, Oriana, please, can you start the recording?
I've started it.
She has. The recording started, it said.
Okay, very well. Good morning. Here we are in today TMS meeting, and we will talk later a little bit, a few minutes before the meeting about an event presented by Dr. Denise Boncioli. She's a particle physicist and a scientist from physics department. Please, Denise, show up and share your slides about an event that's going to happen tomorrow at the University of L'Aquila. Thank you, Denise.
Hi, everyone. Can you see the screen? And
yes,
hear?
Very well. Thank you.
Okay, thanks. So thanks a lot. As Giovanni said, my name is Denise Boncioli. I'm a physicist working at the Department of Physics here in L'Aquila. I just wanted to advertise this event that is going to happen tomorrow. That is the International Day of Women and Girls in Science. This is a day that has been promoted by the United Nations in order to support the access and the participation of women and girls in science. So here in L'Aquila, the University of L'Aquila and the Gran Sasso Science Institute, together with this project for STEM disciplines, we have organized a series of seminars and we had these initiatives with some communicators of science and scientists just to promote and to invite young pupils to know what is the STEM world and how we are passionate about physics and science in general. So we have invited Giuliana Galati. She's a physicist and a communicator of science. She will present what is her experience as young scientist working in Italy. Then there will be a philosopher, Lorenzo Gasparrini. He's going to tell us about why it is important to have a gender balance, to promote gender balance, and that this is not just important for women, but it is important for everybody. We will have also a talk by Serena Giacomin, who is an expert in climate changing, and she is going to talk about this, which we find a topic that is very interesting also for young pupils to whom this event is dedicated. And then we will have also Lucia Botano, who was former director of the Laboratori Nazionali del Gran Sasso, and she will tell us about her experience as the first woman as a director of one of the most important underground laboratory of physics in the world. So you are all invited to attend. This will be in presence at Coppito at the University of L'Aquila. But you can also attend online. The links are the ones you can see here in the picture. So I hope I hope you can enjoy this event, and thanks for letting me advertise this here. Thanks, Giovanni.
Thank you, Denise. Very interesting. So please tomorrow come to this event in Coppito. Now, thank you. Now, I'm going to present the speaker of today. The seminar is entitled Toll-like receptor CD180, an environmental sensor in chronic lymphocytic leukemia, CLL cells. It will be presented by Dr. Nina Porakishvili from University of Westminster, London, and Professor Peter Lydiard, again from University of Westminster, London, and University College in London. Nina is she's a full professor at University of Westminster. She's teaching immunology. She's done a long list of research results, and the most important one is she's going to present today about the demonstration of the heterogeneity of the expression of the toll-like receptor, and many other more research results. She's from Georgia, and she's been having a lot of international experience. And we met actually in one of the European projects, starting from the Tempus time, and now it's called the Erasmus Plus. So I'm glad that these very long friends now can give a talk to us. And then talking about Professor Peter Lydiard, is associate professor at the University of Georgia, Tbilisi, University College in London, visiting professor, and emeritus honorary professor, St. George University in London. Very well known for his book about immunology, and also he was working with many people internationally on the immunology field, and he has a so long list of results that I'm embarrassed to synthesize. So the best way to do it is just to leave them to talk to you about this very interesting talk. So I will leave the word to Professor Lydiard. Please, can you switch on your webcam and start your screen sharing? Thank you.
Okay, so shall I start?
Yes,
Yep.
please.
Okay, so thank you, Giovanni, for that nice introduction. Yeah, we've, as people may know, we've been friends for a long time, and it's in fact many years now, actually, probably about 10 years, I would have thought, from the time we first started working together. So today, then, we're going to talk about chronic lymphocytic leukemia. And I'm glad to see that we have some of our international CLL group who've joined us, which is really nice. And so what I'm going to do is, since I'm the eldest of the group, I'm going to sort of introduce the CLL and introduce you to CD180. So the title then has already been said by Giovanni. So that we're going to talk on this particular talk. We're going to talk specifically about CD180 as an environmental sensor. And first of all, what about chronic lymphocytic leukemia? Well, it's the most common adult leukemia in the Western world. Usually over 58 plus, but as you'll see in a moment, actually it can start much earlier these days, at an earlier age. There's around 3 per 100,000 per year new cases in the UK, and the disease can actually be indolent or progress rapidly, and I think Nina will talk a little later about the prognostic indicators that may help in determining that. Then the incidence of CLL then is very low in the Far East, and this is not completely understood. It may be genetic, partly genetic, and the incidence of CLL is higher in males than females. And this just shows a graph. The blue is the males here, and you can see already it's now, you can start to find CLL quite early, actually, from 40 years on. So it's become a younger disease as well as an older disease. So what are the main biological features then of chronic lymphocytic leukemia? As I've already said, it represents the most frequent B-cell leukemia, particularly in the elderly. It's a clonal disease. Now, majority of lymphoid tumors are in fact clonal diseases. And the cells that you find lots of these in the circulation being a leukemia, and they have a particular set of markers. So they characteristically have a marker called CD5, which originally actually is defined as a T cell marker, but it's on a subset of B cells, of normal B cells. CD19 and CD20 are classic markers of B cells, and all B cells have, to a certain extent, some IgM, except for plasma cells, because they don't need IgM as a receptor for antigen. But CLL has a low expression. and it doesn't proliferate that fast. There's accumulation in G0 to G1 in the body. There's a prolonged cell survival. We don't know completely about the pathogenesis, although we're moving towards that. It can either be indolent or aggressive, and for some time now it's been clear that microenvironment play some role in keeping the tumor going. And what microenvironmental sensors do we have? If we look at this, actually shows CLL cells and what kinds of sensors they have. Particularly, they have the B cell receptor here, which is surface IgM, and that's how it's able. That actually can recognize particular antigens, if you like. And in fact, it's now clear that the majority or many of the CLL cells, their B cell receptor is actually targeting not only microorganisms, but also some self-antigens. So many of the CLL that have been looked at, in fact, we showed this many years ago, that they actually have receptors that can recognize self-antigens. And particularly, I want to focus down here on the toll-like receptors. There are many now of these sensors that are present on B cells, but also on a number of other cells of the body. And these toll-like receptors then, these can recognize particular, as we'll see in a moment, they can recognize particular components, mostly of microorganisms, but also of self-antigens, self-molecules, as it turns out. But the important point is that cells, whether they're any kind of cell, actually has molecules in their surface and in the cytoplasm that enable them to sense their environment, enable them to know where they are, enable them to respond to their environment, and they send signals via a number of different signaling pathways. And Nina will be talking about that in relation to CD180 in a moment. So if we look at the toll-like receptors generally, then they recognize particular TLR2, for example, will recognize particular lipoproteins. Some viruses, TLR5 recognizes flagellin on microorganisms, and so on. So it's fairly well known the ligands, if you like, that attach to these toll-like receptors in order to drive them into sending signals to the nucleus. What do we know? What's 180? What is CD180? Well, we first became interested in this in a moment, and I'll tell you why, which is a kind of introduction to what Nina's going to be talking about. CD180 then is a type 1 transmembrane protein. It has a molecular mass of around 155 kilodaltons, and it was initially discovered in mice, actually, by a molecule called RP105. And this subsequently was identified as a molecule CD180. It was called CD180, which is present also in humans. And it's made up of around 661 amino acids. Why I showed you TLRs is because CD180 has a very high degree of sequence homology and structural homology with the toll-like receptors, and particularly toll-like receptor 4. And like those, if you look at the structure, it has extracellular leucine repeat units, which are involved in the way in which the molecule sits outside the membrane. Unlike the toll-like receptors that I've been defining, CD180 doesn't have a true ligand. It's what we call an orphan receptor. And what's interesting is there's lots of ideas about it, what the ligand is, and some suggestions. The most common suggestion is, like TLR4, part of it attaches to endotoxin, lipopolysaccharide of the gram-negative organisms. So lastly, then, it's CD180 is present on B cells, dendritic cells, and macrophages of the immune system. And these are, of course, antigen-presenting cells. And this is obviously telling us something about CD180 being virtually exclusive on these type of cells. So what gave us the idea that CD180 could be important in CLL cells? Well, there was an observation made by, in fact, one of our collaborators, Ed Clark, with other people in the States. And what they showed was that if you took murine B cells, if you added anti-CD180 to them and then added anti-IgM, which is the trigger, if you like, once the cell becomes activated recognizing an antigen, in this case the cells went into apoptosis. Without CD180 pretreatment, there was no apoptosis. In fact, it activated the cell. So then the question became, if this is true, maybe we could use this in terms of CLL that have CD180, as we discovered on, and had IgM on their surface. Then maybe we could use this as in a form of treatment and induce CLL cells to apoptose and therefore die. So it may be a kind of treatment. So that, together with the idea that that CD180 has a sensor activity, really made us very interested in this molecule. Okay, so thank you. I'll move on now to Nina, and we'll take questions at the end. So thank you.
If you could stop sharing, please.
Yep.
Thank you. So now I'm going to do that. And we're going to get from slide. Okay. So here we are. What is important for CLL also, why we've got so much interested, Peter kind of laid a very important foundation for the rest of our talk, is that it is highly heterogeneous disease. Highly heterogeneous in the course of the disease, and therefore sometimes we even say that each patient comes with their own disease. They're so different from each other, and they're different because of the so-called prognostic indicators and the biology of CLL cells. These prognostic indicators are the molecules that are expressed on the surface of the CLL cells, of leukemic cells, or perhaps they are signaling molecules such as tyrosine kinases. They are very important genetic chromosomal changes that are associated with the different course of the disease. So therefore, that makes it very difficult to approach, very difficult to manage, and very difficult to treat. So we were thinking to start with, as Peter said, whether we can use the CD180 for the triggering of apoptosis of CLL cells, because that's what was shown in mice. However, we also were interested to see how the CD180 will get into this picture of the various prognostic indicators. I listed prognostic indicators here, but they're not, unless you work in the field, not important to remember, but I will be getting back to them when they have any possible connection with CD180. So the first thing to do was really to find out, because CLL is such a heterogeneous disease, whether CD180 is at all expressed by CLL cells. We didn't know. When we started the work, we started really from scratch. So therefore, that's our first paper, which was published in 2005. And what we found there is that you can see the white columns are the control normal B cells from control individuals. And you can see that we were looking into the overall B lymphocytes, as well as CD5 positive B lymphocytes, because CLL is a leukemia of CD5 positive cells. Those of you who might work in the hematology would know that there are CD5 negative and CD5 positive B cells. So we just checked. And all normal controls B cells expressed, as expected, high level of CD180. However, in the CLL, we found that 2/3 of the patients, and funnily enough, everything comes like 2/3 in CLL, 2/3 positive for this, 2/3 positive for that. And there were positive, 2/3 were positive for CD180. And you see that the expression level was lower than that of the normal control B cells, but 1/3 of CLL cells didn't express CD180 at all. So that was a very important the finding. The next finding, which was published in the same paper, was, okay, that's negativity and positivity for CD180. Is it prolonged in time? Maybe we just captured the CLL cells at the stage when they were negative or positive. However, we observed the CLL cells from individual patients when it was easier to have the ethics, by the way, at up to the 24 months. And you can see that the positive patients remained positive, or the cells rather from the positive patients, negative remained negative. So therefore, this positivity for CD180 or negativity for CD180 is an intrinsic the feature of CLL cells from particular patients. Now we come to this one of the important prognostic indicator, which is still a part of the clinical practice, looking into the special genes that are coding for B cell receptor for IgM, IgVH genes, and they are the mutated versus unmutated. Now, because normally we think mutated is bad and unmutated is good, in fact here, mutated genes for IgVH means that a B cell is undergoing normal development, because under the normal development, those genes should mutate. Therefore, unmutated are abnormal entirely, let's say in a more simplistic way, mutated are good. But what we saw is that the CD180 is expressed mostly on the patients which had mutated CLL, which again, I emphasize, is a good prognostic indicator and still in clinical practice compared to unmutated cases. And that was very interesting, and I'll tell you in a moment why it was interesting. So therefore, we published it for the first time. It has been established that CD180 is mostly expressed on so-called MCLL, which have better prognostic future, let's say, and the indulgence of the disease compared to the unmutated ones. So the next question which we asked was obviously, okay, it is expressed on 2/3 of the CLL patients, but is it functional at all? So again, in the same paper, we were looking for the two readouts for the function of B cells. We ligated, because as Peter said, this is an orphan receptor, so we don't have a ligand. We could only use antibodies to engage this receptor. So we used the anti-CD180 antibodies and looked whether what happens with the cells, whether they get activated and whether they get into the cellular cycle, proliferating. Activation was we measured with CD86, which is B72 molecule, and the proliferation we measured with Ki67, one of the proteins associated with cellular cycle, or cyclin, if you wish. This is a picture for normal control B cells. It's interesting that I would rather think this is an artifact than perhaps the experimental error, but they are uniformly, the normal B cells responded to CD180 ligation by activation and the proliferation. And so did now what happens, we have 2/3 positive for CD180, 1/3 negative. Out of those 2/3 that were positive, half responded in a manner that was very similar to normal controls, and half didn't respond at all. So now we have 3 groups of CLL patients, but like if 2 wasn't enough. So we have the CD180 positive responders, we call them responders, CD180 positive non-responders, and we have CD180 negative. So everything comes in 3 in CLL, as we usually joke. Now, what also happened was that apart from the responses such as proliferation, activation, not surprisingly, CD180 protected the CLL cells from apoptosis, and that was unexpected. So what we are measuring here are the apoptotic cells, which are the dim for the on the left, dim for the DIOC6, which is a mitochondrial dye, and the bright cells that are the viable cells. Now, if you take, but it's an interesting feature of CLL cells, if you take them out of in vivo, out of the patient, and culture them, they die quite often, quite quickly. So therefore, you can see spontaneously, we have a lot of dead cells here and very few viable cells. But if we engage CD180, we actually have the number of the viable cells significantly increasing from 27% to 61%. Now that was quite unexpected because if CD180 is associated with good prognostic group, mutated CLL, how come it protects CLL cells from apoptosis? Yeah, so it just kind of didn't work, wasn't very logical. So these are the conclusions here, which I already mentioned, so I'll just go on. Therefore, the next, we had 2 papers published. One, I didn't show the picture in 2011, and the second in 2015 in molecular medicine, again with our international group, where we actually started to look what's going inside a CLL cell. Okay, where they are proliferating, they are activated, they're protected from apoptosis via CD180, but what kind of signals we can identify there? And the next slide, which is one of the most important slides actually, on which we base our current data and our current research, it's a little bit here busy, I would say, but I will try to explain. So therefore, what we realized was that we couldn't have looked into all protein kinases or all the signaling pathways. That wasn't possible. So we choose 2 signaling pathways, one through the protein kinase called AKT, and the second through protein kinase called p38 MAP kinase. Now, with MAP kinase, it wasn't very clear, but AKT was really very strongly associated with the survival of the cells. And this is a part of the BCR, B cell receptor signaling. PI3 kinase, the AKT, is a link which provides survival. So therefore, because CD180 was providing survival from apoptosis, we were interested to look into that. As an alternative, we also used p38 MAP kinase. What turned out is that we actually found that very often CLL cells, after being treated with anti-CD180 antibodies, showed express alternative signaling patterns, either AKT or p38 MAP kinase. So we can see here the signaling, and we measure signaling via phosphorylated versions of protein kinases. So you can see this is for AKT on the top. On the bottom, we have the information on p38 MAP kinase. Look at the control cells here and here. Do you see my cursor, by the way? Sorry for asking so late. Thanks. So you can see that the control cells respond by phosphorylation of both. So control cells are like the dual. They do AKT, they do p38 MAP kinase. And if you see here, we have a small number of CLL cells that follow the pathway, if you wish, of the normal control B cells. They phosphorylate both of the protein kinases. However, in the vast majority of cases, we had alternative phosphorylation of AKT and p38 activity drops, or we have the phosphorylation of p38 MAP kinase while the AKT drops. So that it looks like there were 2 alternative pathways. And of course, not surprisingly for such a heterogeneous disease, we had some of the cases which were non-responders. So, well, at least those 2 protein kinases have been phosphorylated. So that was the key for our moving towards the idea, what can we do can we at all kill the CLL cells using CD180? And that became more and more problematic because we were seeing that CD180 actually can trigger the pro-survival AKT pathway. So then we looked into what those pathways really do. Not surprisingly, we saw, it's a very busy slide in here, so I will ask you to read the paper if you are particularly interested. I'll just tell you in verbally and then concentrate on this particular picture only. So what we saw that indeed, if the AKT was phosphorylated in CLL cells, they were surviving from apoptosis. If, however, p38 MAP kinase was phosphorylated in our hands, that led to the apoptosis. So that gave us some hope. Okay, we can still trigger apoptosis of CLL cells. Now, what will happen if we combine, as Peter said, CD180 with IgM? And this is shown in here. So this is a spontaneous the activation of AKT protein kinase. This is after we use anti-IgM, so when we induced IgM signaling, and IgM signaling in CLL cells is very well known. It's leading to the survival. So not surprisingly, then AKT went up. However, when we pretreated the cells with CD180 and then came with BCR, it returned back almost, AKT phosphorylation returned almost to the basal level. So indeed, CD180, interestingly, despite on its own being able to induce survival of CLL cells, when combined with BCR, exactly what people have seen with murine cells. When combined, or both are combined, actually the survival drops. And that was also shown for the when we measured apoptosis. Just to show you a very interesting picture in here, which I actually, have done these experiments in the Feinstein Institute in New York, in the Nick Kuratz's lab, who is one of the major players in the field of CLL, as Peter is. So you can see here the two of the protein kinases, ZAP76, which I'm not showing the data, AKT, IgM shows significant phosphorylation. The red CD180 shows low phosphorylation. When combined together, CD180 is pulling IgM down. So therefore is blocking the IgM-mediated signaling. And in the same paper, we came up with this picture. Interestingly, you see unknown ligand. CD180 can signal via the pro-survival, the pathway, using BTK, which is Bruton tyrosine kinase. And I didn't show the data, but we've shown that BTK is linked with PI3 kinase, is linked with AKT. This all leads to the upregulation of anti-apoptotic proteins, MCL1, BCL-XL. That was all done in this paper, leading to the survival. In some patients, CD180 engagement does exactly that. In other patients, it actually activates alternative pathway, P38 MAP kinase, leading to apoptosis. But when you combine the two, IgM signaling is switched to the P38 MAP kinase inducing apoptosis. So this hypothesis of ours worked, that you can actually engage CD180 first, then come up with IgM and kill some or the significant bulk of CLL cells. Now, what we are doing these days, and I will skip a little bit just to show in this picture, there's another B cell receptor, not only IgM, but also IgD, which is quite an enigmatic B cell receptor because people don't know much about it. I mean, we pretend that we do know. I teach my students that the IgD is the negative regulator, very much likely, of IgM, but it is expressed at the same level of IgM, but its involvement in intracellular signaling is always very interesting and, as I said, enigmatic. So what IgM does at the same time, if we think about the situation in vivo, as Peter showed, there are a lot of the antigens floating around, interacting with IgM as well as IgD. And we also think that there is unknown ligand for CD180. In vitro, we used CD180 first, then came with IgM. But would it happen in vivo? Would they wait for each other? Or they will be engaged either simultaneously or in a different order. So therefore, what we are doing now, and we have our Westminster group here present with the Dr. Emanuela Volpi and Curtis Edwards, who is now driving. He's now the doctoral researcher. He's a master, but now he's a doctoral researcher. And he's driving this research in the Westminster at the moment. And also Tamune Tsertswadze from Georgia, who is driving the CLL research in Tbilisi State University. So we're now interested in looking into the interaction between CD180 and IgD, not just only IgM, seeing how IgD might interfere with this very nicely set up the scheme of ours. So again, I will skip the conclusions. You will have them in the presentation. So it's just repeating what I've just said. Now, how Curtis started his studies in the Westminster was that we were going further deep into tissues, if you wish, because what has been done so far, I haven't mentioned it, was done on the peripheral blood. But really, in peripheral blood, immune cells and CLL cells already traffic. They just go around from one lymphoid site to another lymphoid site. And mostly they are resident in the lymphoid tissues and organs such as spleen, lymph nodes, and other mucosa-associated, so on. For CLL, it is well known that they are trafficking quite actively from circulation into the lymph nodes and back again. And when they get into the lymph nodes and bone marrow also, which I haven't mentioned, there is when they proliferate. So they don't proliferate in the peripheral blood because they are traveling with such a high speed. But in the lymph nodes, supported by the microenvironment again, in the bone marrow supported by microenvironment, they indeed proliferate. And there's a name for those pieces of the tissue called proliferation centers. So what Curtis did, and that was published last year, he looked into the data which was available by the collaborators of ours from Queen Mary. There were paraffin sections from the lymph nodes and the tonsils as controls, normal human tonsils, first of all, with CD180 expression. And we saw exactly the same as we saw with peripheral blood, heterogeneous expression. If you look at the picture here, which is kind of brownish, these are the normal controls tonsils, and you can see that the normal B cells expressing CD180 are kind of grouped together in a special zone. However, if you look at the CLL, we have negative to the right here, the intermediate and positive in the expression for CD180, the same heterogeneity. What Curtis also showed was that this CD180 in the lymph nodes is again associated with Ki67. So the cells that are positive for CD180 are preferably proliferating. So that supports what we saw in the peripheral blood. And very importantly, this is a heterogeneity showing that we have really very homogeneous expression of CD180 on control B cells, but heterogeneous in CLL. And what Curtis further shown was that, first of all, there is what we haven't mentioned, Peter and I, apart from the all those prognostic indicators, as any disease, this has a stage, so that's staging. Binet staging and Rai staging. In Europe, it's a French discoverer of CLL, and there is an American of Indian origin discoverer, Kanti Rai. So in the US, we use the Rai system. In Europe, we use Binet system. So the more advanced the stage is, the more severe is the disease. And what Curtis has shown is that you can see the Binet stage, that CD180 is associated more with the earlier stages. So it seems that CD180 is a kind of an earmarking the CLL cells which are not very aggressively developing. And the most important picture you can see here on the right is that for the first time, actually, Curtis was able to show that the survival of the patients at the blue line, whose cells are positive for CD180 in the lymph nodes, is considerably higher than survival of the patients without CD180. So therefore, CD180 seems to be favoring and somehow perhaps affecting and influencing the indolent course of the disease. And that was one of the most important points that we were making. So, okay, so where we are at the moment? At the moment, we are doing experiments on the cell lines and CLL, trying to combine IgM, IgD, CD180, and another player that appeared quite recently and was actually interestingly researched together with Ed Clark, who is a father of CD180, we call him father, he discovered CD180, together with a Ukrainian group. There's a SLAM receptor called CD150, which is also interacting with CD180. So we not only have 3 players now, but 4 players: IgM, IgD, CD180, CD150, and in different combinations, we want to see how these receptors, what really they do, which the signaling they induce, and what actually happens after the signaling events. Are the cells dying, protected, and how this is all linked to the with the clinical information that we can get from the patients. And the second question that we're asking, and that actually where Tamuna also comes from Georgia, actually Tamuna works on those 4 receptors as well, Tamar Tsertswadze, and she's also just literally last week sent some very interesting data that we want to incorporate here. The second question that we're asking is, what is the reason for heterogeneity? Why all normal B cells express CD180, and in CLL we have low expressors, no expressors, and so on? Could it be that this is due to the RNA, the levels? Could it be that there is a gene mutation and therefore the message is not produced enough? And now what we have a paper in preparation, was again led by Curtis. What we've done, we've had the data generated in our group, and also the data available, metadata available from the databases. And what we saw really is there is no difference whatsoever in the expression of RNA for CD180, not only between normal controls and CLL, but across CLL, the bone marrow, lymph nodes, or peripheral blood. So therefore, RNA is not guilty for not expressing CD180. What could be guilty then? We have now 2 possible culprits. One could be that there's a problem with the post-translational, perhaps modification of CD180, post-translational events, maybe interaction with the Golgi and the transport of the CD180 on the surface. And in this case, we should see accumulation of free CD180 inside the cells. Or it could be that the problem is with this MD1 molecule. I'll show you in a moment. So MD1 molecule is a small satellite for CD180, which is not a transmembrane molecule. You can see here, very similar to toll-like receptor 4 that has a similar, the satellite MD2. And we always knew that without MD1, the CD180 will not be expressed on the surface. So there are 2 possibilities. MD1 is a limiting factor, or there's a problem with the transport to the surface. And at the moment where we are, we actually saw that the MD1 RNA expression in CLL is indeed lower than in control cells. So that could be the reason. And we are now, we are buying in the process of antibodies to MD1 to screen our, the CLL cells for MD1 expression. And the second, really a very fresh, you can see it's paper in preparation, very fresh data showing that indeed, if we look at the CD180 expression on the surface and intracellularly, it is almost always higher intracellularly. So whatever we have on the surface, even in CD180 positive cases, a lot of the molecule, a lot of CD180 in CLL cells is actually stored inside the cytoplasm. So therefore, at the moment, we are looking for the questions. What is more practical? How these 4 receptors can show us the way, pave the way towards the management of chronic lymphocytic leukemia by engaging CD180. And second, if you wish, fundamental biological question, why CD180 is not expressed uniformly by CLL cells as the normal control B cells do. So thank you. I finished. These are the acknowledgements led by Curtis and the whole range of our previous PhD students, Uzma Said, Ketki Vispute, Nadika Rajakaruna, who contributed a lot to this project. Kristina Zaitseva, our alumna from, she was an undergraduate student, but really put a lot of work into the lymph node paper. Dr. Tamar Tsertswadze from Georgia, and the heavyweight contributors, if you wish. Professor Amit Natwani, our the consultant at UCL, sorry, yes, Royal Free and UCL. Then Professor Ed Clark, formerly from the University of Seattle, father of CD180, Nicholas Kiorazzi from Feinstein Institute, USA, Dr. Sergei Krysov, our kind of new collaborator at Queen Mary, Dr. Emanuela Volpi here present. I didn't show the data because we want to use it first, but one of the, as you can see, the future studies here is also to see the relationship between CD180 and prognostic indicators. And major prognostic indicator now in the world of CLL are chromosomal aberrations. So we're looking how everything we're looking here correlates with the mutation or deletion of the TP53 and other the chromosomal aberrations that are considered to be prognostically favorable. So Dr. Manuela comes with this huge the expertise in the field of genetics, and she's also interested in chromosomal instability, or rather genomic instability. So looking into the correlation between genomic instability and CD180 story. And here the humble, well, not humble Professor Peter Lizard, and humble myself at the end. Thank you.
Thank you, Nina and Peter. Very interesting talk, really interesting. I'm ignorant of all of this science. Definitely, I have many questions, but let's see from the audience, there are more specific questions from people that does biotechnology, biology, microbiology. So please show up, talk, or just put in the chat your question. I can read it.
Would you like me to stop sharing or keeping the slides in case if someone is asking a question?
It would be nice to keep sharing if people ask questions about pictures. Actually, I was impressed by this powerful protein. It's a protein, right? So
yes.
It could be used for prognosis and also for treatment, right? Is that correct?
Prognosis, definitely, because we were always looking to the finding really correlation between patient survival and CD180. Finally, we did. We are not sure whether this could be also found for the peripheral blood CD180 expression, which should be easier for prognosis, of course. And linking it to the chromosomal aberrations, of course, will increase its prognostic value. However, because CD180, as Peter said, is expressed by antigen-presenting cells, we can put forward the idea of, so therefore there will be limited response to CD180 if we wish, but we need to avoid something like cytokine storm, for example, if we really induce the CD180 in vivo. But the idea is if we apply anti-CD180 in vivo, then it could be that we will be triggering the apoptosis of CLL cells, but normal B cells might follow the situation as well. But that happens with any immunotherapy. Rituximab anti-CD20 is inducing apoptosis of leukemic cells, rheumatoid, the autoantibodies producing B cells, as well as normal B cells. So that's always a side effect.
Great, thank you. More questions from the audience, please?
Sorry, I kind of jumped into answering. This is a feature of the lecturers. You know, maybe Peter would like to add something. Sorry.
Well, I actually have a question for Peter because I remember dendritic cells. Is that correct?
Yes.
So is that something related to the brain? What's the role of the brain here?
Well, it's a good question. I mean, generally, there's a lot of interest, in fact, in the brain now in actually controlling immune responses, and the relationship that it has. Dendritic cells. I mean, the thing we talk about with dendritic cells is they're like really nerve cells in terms of their dendrites. If you think of a dendritic cell, the idea of a dendritic cell is that it's got lots of these processes, just like the end of a synapse of a nerve ending. And the idea is with dendritic cells is having those different dendrites, if you like, it enables a large number of cells to get cozy with the cell and to interact with the sensor receptors that we're talking about essentially. So that's really the link between at this stage between the brain and dendritic cells.
Sounds great. Thank you. Thank you. More questions, please. Don't be shy. We have quite few PhD students from our department, also from our other universities here listening. So please, Anna, go ahead. Professor Anna Teti.
Hello, yes, I'm not a PhD student, unfortunately. I'm not expert in this topic as well. However, I work since many years on cells that now it's clear that it's part of the immune system, and it's an antigen-presenting cells. They are the osteoclasts. That are cells that resorb bone, so stay in the bone marrow, and take part to the innate immunity, presenting the antigen, and also with immunomodulator activity. I wonder if you have any information on their role in this disease, or if there are no roles at all.
I will answer very shortly. Maybe Peter can have having more information about that. As for CD180 expression, I don't think anyone looked into osteoclasts. Maybe Curtis is here and he did a lot of literature search. Maybe he can, in case if he found somewhere osteoclasts expressing CD180, would be interesting. But on the other hand, because it's not for a chance that antigen-presenting cells are expressing CD180, so therefore there is a theoretical possibility, at least if not practical, already done, by the way, that CD180 is expressed by osteoclasts. Generally, the osteoclast involvement with the CLL as a disease. Peter, would you like to say something about this?
No. As a disease, I don't think there's any relationship with, obviously, with bone resorption or whatever in CLL, but I think osteoclasts are very interesting, and they clearly are regulated because of the basically osteoclasts regulate the osteoblasts, because osteoblasts are related to producing bone. Osteoclasts are related to resorbing it. So what's interesting is that, and I don't know the literature on this, but whether osteoclasts have very many sensors. For example, do they have any immunological sensors that we know about? And you're saying they do, which is very interesting, because it might mean, and they may have cytokine receptors, so they may be regulated themselves to change bone, to change the relationship, the balance between osteoclasts and osteoblasts. So I think it's a fascinating area.
I think that if I may just add, if CD180 is expressed by osteoclasts, being partially macrophages, former macrophages, as monocytes, let's say, which develop into osteoclasts. In this case, could be that we can drive the proliferation and the bone growth using the CD180. Sorry.
No, you're absolutely right. What you said is absolutely correct. It seems, however, that immune osteoclasts resorb less the bone, so it seems that they are implicated in activities that are not completely related to bone resorption. And another information is that they are implicated in multiple myeloma development, which makes sense because the multiple myeloma needs room in the bone marrow to develop, and the osteoclasts are ideal cells to make room there. But it would be interesting to expand a little bit also on leukemia.
Can I
Some
just
of
ask about
the myeloma cells express CD180 as well. Yeah. So Peter.
I think it's interesting. So there's a subset of osteoclasts that you talk about that are so-called immune.
Yes.
So it'd be very interesting to know whether they're CD180 positive.
This I don't know. I don't know, but I can provide you some articles on that.
Okay.
Maybe Curtis can answer this question because his hand is up. I can see.
Hi everyone. Yeah, so the only thing that kind of springs to mind in terms of osteoblasts, osteoclasts, is there was a paper in last year in 2021, and this was a retrospective study of mRNA analysis data, gene expression data. And interestingly, this group actually found that CD180, along with two other genes, was actually formed a model, a prognostic model for more severe osteoblastoma. And interestingly, which kind of relates to our studies, they also found that when CD180 was increased in terms of gene expression, that actually painted a more positive prognostic picture. So it's kind of curious because we found that in these B cells, these CLL cells, that we don't see a change at the RNA level, but actually some other groups in different cancers have found that actually the changes in the gene expression are important. So hopefully you'll be able to read this all in our upcoming review.
Okay, thank
You have
you.
to show your face.
Right.
Oh, okay.
Hi everyone.
Thank you,
Sorry, I'm in the
Curtis.
lab.
Thank you,
Ah,
thank
in
you.
the lab.
Very, very interesting interaction here. It's going on. I may have a last question if nobody else has one. Since some of my colleagues are involved in bioinformatics, I'm wondering if there are any public data set available where this protein expression can be correlated with other biosignals or proteins or any other kind of data.
One of the data for the RNA expression shown in here, yeah, the metadata which Curtis used for the analysis for RNA expression.
Thank you, thank you.
There are plenty. So and therefore, yes, this is available, and yeah, we are very interested in metadata analysis, really. Because, well, the coronavirus brought it into our lives, but it actually did a good job.
Yeah.
Because, you know, otherwise data was lost somewhere. People were not using what other people have done.
Right, right. Yeah, data analysis and discovery, data mining, this kind of stuff. Right.
Absolutely.
It'll be interesting to involve some computer science student to do that.
Yeah.
Yeah.
Mm-hmm.
Okay.
We can discuss that with pleasure. Yes, why not?
Why not? Yes, why not? Definitely. Any more questions, please, from the audience? We have a few more minutes. If not, that's fine. I really enjoyed this talk, and thank you again, Nina and Peter, and all of the people from the audience, Professor Nateti, Curtis, and many more that participated and asked questions. So maybe we can have another one on your next progress of research. It would be nice to invite you again. And so,
thank you so much for inviting us. It
Yes,
was really great
thank you.
pleasure meeting all
Thank
of
you,
you and
Giovanni.
sharing our experience.
Yeah, it's
Thanks
been
a lot.
thank you, and goodbye. Take
Bye-bye.
care, and have a nice day, everybody.
Okay.
Thank
Bye-bye,
you.
Thank you.
everybody.
Bye-bye.
Bye,
Bye-bye.
friends in Georgia. Bye.
Ciao.
Bye.
Ciao.
You know, bye.
Bye-bye.